Hi, good afternoon. I'm excited to be a part of this year's Generic Drug Science and Research Initiatives public workshop. For those who aren't familiar with me, my name is Irene Chan, and I'm the Deputy Director in the Office of Medication Error Prevention and Risk Management. So today I'll be speaking to you more about comparative use human factor studies for ANDA products. Just a quick disclaimer, for work prepared by U.S. government employees representing their agencies, there is no copyright and these work products can be reproduced freely. Reference to any marketed products is for illustrative purposes only and does not constitute endorsement by the U.S. government, the Department of Health and Human Services, or the Food and Drug Administration. The objectives for this session are to describe what the objective of a comparative use human factors study is. We'll review the steps involved in designing a CUHF study. We'll present an example of a hypothetical CUHF study, and then we'll review tips for submitting a CUHF protocol. A lot of the content that we'll be going over today is reflected in the draft guidance shown on the screen here. This particular guidance focuses on the analysis of the proposed user interface for the generic drug device combination product when compared to the user interface for the reference listed drug, or the RLD. The guidance provides a process overview that starts with comparative analyses, or threshold analyses as noted in the guidance. Now, after comparative analyses are done, as a sponsor you need to determine were there any differences identified. If the answer to that question is yes, then you'll also have to take the step of determining whether the differences were minor. Now, if the differences were not minor, or these were other design differences as noted in the guidance, then this is where we would encourage you to have further discussions with the agency. We would also need to determine whether additional information and/or data, such as data from a comparative use human factors study or CUHF study, may be warranted. Remember, ANDAs rely on FDA's finding of safety and effectiveness for their reference listed drug. An ANDA requires demonstration of sameness of a number of characteristics plus additional information to permit reliance. Generic combination products classified as therapeutically equivalent to the reference listed drug can be expected to produce the same clinical effect and safety profile as the RLD under the conditions specified in labeling. So what does that really mean? Well, it means that you're not establishing new safety and efficacy for the proposed generic product. Generic products are essentially confirming sameness to the reference listed drug. Therefore, we need to discuss a comparative approach. For a CUHF study, the objective is to demonstrate that differences would not preclude approval of the proposed product in an ANDA. Generally, these are going to be simulated use studies, and generally, a non-inferiority study design will be appropriate. The goal is to show that the patient experience using the generic combination product will be no worse than that with the reference listed drug, with some allowance for random variation. So what steps do you need to take as the sponsor? First off, you're going to want to identify who your users will be. FDA's focus is on whether substitution can occur with a full expectation that the generic product will produce the same clinical effect and safety profile. Therefore, you should include current end users of the RLD. You should consider if your analyses indicate that specific subpopulation should be the focus of a study. And you can consider whether a difference in design may impact critical task performance for patients diagnosed with certain indications only. In any case, you will want to have some discussion with the agency to ensure appropriateness of the end users that are recruited. Secondly, you'll want to identify your delta. You should consider if there is existing literature or data that provides a baseline knowledge about use error rates for the RLD for the critical tasks of interest. You need to demonstrate that the error rate with the generic product is no greater than the error rate with the reference listed drug plus delta, where delta is an acceptable deviation above the error rate for the reference listed drug. Delta should take into account that there is some allowance for random variance with the error rate for the reference listed drug that is expected. Delta should take into account the risk that any difference in outcome will pose to the patient. In other words, what is the clinical consequence associated with a critical task failure? Be prepared to justify how you derive delta in your submission. Then you want to decide on paired design or parallel design to your non-inferiority study. A paired design will generally be applicable and more efficient with respect to resources. Subjects should be randomly assigned to the sequence of use, such as AB or BA, to control for order effects. In a paired study design, each subject is his or her own control. With this type of study, the sample size is often smaller than that required for a parallel design. First, you would enroll subjects, then you would randomly assign each subject to one treatment, and then the subjects would receive the treatment not previously assigned before you ultimately analyze the data. The analysis must consider correlation within subjects because success rates in the two treatment groups are not independent. A parallel study design, on the other hand, usually requires larger sample sizes than a paired study design. In this case, you would enroll subjects and randomly assign each subject to one or the other treatment. Either the subject will get the reference listed drug or they would get the generic product. And then ultimately you would analyze the data. Statistical tests with this design are often more straightforward than for the paired design. Afterwards, you want to calculate your study sample size considering assumed error rates and delta. Please keep in mind that the flip side of error rates are success rates, and this will become more important as we talk a little bit later. Typically, the acceptable type 1 error probability, or alpha, will be set at 5%. What you're watching for are type 1 errors where you reject a true null hypothesis, or type 2 errors where you have non-rejection of a false null hypothesis. Ultimately, you want to consult your statisticians. This is extremely important. You need to have the right expertise to undertake this on your behalf so that you can ultimately ensure that the study that you design will in fact allow you to draw the conclusions that you're hoping to make. Next, you would submit your study protocol to the FDA and get feedback before initiating a CUHF study. This can be done via control correspondence or pre-ANDA meeting. It is worth underscoring that you should wait to get the feedback before you proceed with your study. Once you have agreed upon protocol with the FDA, then you'll proceed with conducting the study. And during the study, you're going to observe error rates and success rates for the critical tasks. When observing the study, you can assign a binary value, 0 or 1, to users for each critical task performed, where 1 is assigned to successful task completion and 0 is assigned to task failures. Then you're going to perform your statistical hypothesis test, comparing the upper bound of the appropriate level of confidence interval for the difference in event rates to delta. So your null hypothesis would be that the error rate for the generic product minus the error rate for the reference listed drug would be greater or equal to delta. Your alternate hypothesis is that the error rate for the generic product minus the error rate for the reference listed drug is less than delta. Rejecting the null hypothesis in favor of the alternative hypothesis supports the claim of non-inferiority as defined by delta. Alternatively, if the study design is based on success rate, then you would perform your statistical hypothesis test based on the following. Your null hypothesis would be that the success rate with the reference listed drug minus the success rate with the generic product would be greater or equal to delta. Your alternative hypothesis is that the success with the reference listed drug minus the success with the generic is less than delta. In this case, rejecting the null hypothesis in favor of the alternative hypothesis supports the claim of non-inferiority as defined by delta. So let's walk through a hypothetical example. Your reference listed drug is an emergency use product marketed as a prefilled syringe with a cap that snaps off. Your generic proposes a prefilled syringe that has a cap that threads off. So here you would require the user to twist the cap off. In terms of the threshold analyses, it was determined that one other than minor difference exists, and that is with removal of the cap. For this example, we assume that cap removal is a critical task. So we would consider that intended users may encounter more difficulty with twisting off the cap, and in a substitution scenario, they're likely to try to snap the cap off, as that is what they're accustomed to doing with the reference listed drug. So as we dive deeper into this hypothetical example, let's think about our endpoints here. What we're focused on is the task of cap removal. Specifically, we want to understand whether patients are going to encounter difficulties and be unsuccessful at removing that cap. Each subject would operate both devices. In this case, we're using a paired design for our study. We would randomize the order within subjects, and other details would be put in place as appropriate, such as masking the devices. In terms of the test then, your null hypothesis is to understand what % of those failing the goal, in this case removing the cap, occurs with those using the generic product, minus the % that are unable to remove the cap with the reference listed drug. Because in this example we set delta at 10%, then the null hypothesis would indicate that based on this subtraction, you would fall greater or equal to 10%. The alternative hypothesis would be that you would fall at less than 10%. So in this example, your sample size of approximately 50 would be determined based on an assumption that 90% of subjects are able to correctly remove the cap. Let's pretend there's information in the literature. You're running this study at 80% power. You want a type 1 error probability of no more than 5%, and your within subject correlation is set at 0.9. So here's an example of what the analysis might look like. If you look at this particular table, you can see the attempts that were both successful and unsuccessful with the RLD, as well as with the test product or the generic product. S here stands for successful and U stands for unsuccessful. So when we look at the outcome then of this study, we see that the difference, in fact, does fall at less than delta, with delta being set at 10%. The upper bound of the 90% confidence interval is less than the 10% margin, which rules out a difference of greater than 10% with 95% confidence. So this is like doing a one-sided test at the 0.05 level. Let's take the same example though, and let's run this in an alternative way. Again, we're focused on the task of cap removal, but here we're going to look at the successes associated with the removal of the cap. Again, both subjects operate both devices. It's a paired study design with randomized order within subject. Other details are put in place as appropriate. And here we have listed the null hypothesis and the alternative hypothesis. Here, when we look at the % that were successful with the reference listed drug and subtract out those that were successful with using the test product, if it's greater or equal to 10%, then that would be our null hypothesis. If it falls at less than 10%, then that's our alternative hypothesis. So again, let's look at the example analysis here. S stands for successful attempts, U stands for unsuccessful attempts, just like we previously saw. The users both use the reference listed drug as well as the generic product. And based on the outcome that you see, especially pay attention to those numbers that are bolded, we determined that the upper bound of the 90% confidence interval is less than the 10% margin based on the difference. This rules out a difference of greater than 10% with 95% confidence. Therefore, rejecting the null hypothesis in favor of the alternative hypothesis supports the claim of non-inferiority as defined by delta. So let's end with some tips for submitting your CUHF protocol. Firstly, clearly identify the user interface design differences. You want to include your threshold or comparative analyses as part of your submission, and you want to make sure that you've clearly articulated where those design differences exist. You want to ensure you recruit appropriate expertise to inform your statistical analysis plan. In other words, make sure you have run your test by a statistician. Within your submission, you explain how you derive delta. It's very helpful to reviewers when you provide samples of your product. It helps the reviewer to visualize the differences that you point out in your threshold analysis. I would also refer you to additional information available in a draft guidance that we have titled The Contents of a Complete Submission for Threshold Analyses and Human Factor Submissions to Drug and Biologic Applications. And last, please wait on agency advice before you proceed with your study. Thank you for your attention today.